lot, Nathalie, for coming.
It's
in
my
spite you
pleasure.
were involved in many other activities of the meeting.
It's my pleasure to be here, and thank you very much for this kind invitation and for being here. I'm really very sorry not to be able to tell you this in Spanish, but it's really impossible for me. I'm really happy to be there. I must say I do this kind of presentation in France for families with leukodystrophies, and I think it's a very important exchange we can have, and so I really hope that you will understand what I say, and if you think not, Juan will go through the slides again so you can understand. The other thing I want to say is I'm very impressed by what has shown Fatima. I have put much less slides on very scientific details. I wanted more to tell you the whole story of this gene therapy approach for this very severe disease, show you how long it is, but how it can work and how it can help for future clinical trials to move forward more rapidly, and what are the things that are important. So I hope this will fit with what you want to hear about gene therapy. So I'm going to tell you about a disease with a very complicated name, adrenoleukodystrophy. And leukodystrophy is, it comes from the Greek language. It means disease of the white matter. The white matter of the brain is the myelin, and that is this thing in white that you see here that is destroyed. The destruction is what you see here in white, going with time. This is one child, and this is different time points, 1, 2, 3, and 4, and in something like 18 months, and you see the progression, the terrible progression of this destruction of the white matter. So this is a leukodystrophy. And what is it actually? The myelin is this sheet around the axon of the neuron. This is one neuron in your brain, and this is the axon. This is what gives you the information to do all your things you do, of course, to speak, to walk, to do whatever you do in your life. And because of this sheet of myelin, this neuronal influx can move forward to your body. If this myelin sheet is destroyed, you cannot do anything more, and finally this axon will die, and finally all the neuron will die. So leukodystrophy is the disease that destroys this myelin sheet in the brain. So you see here how it can be destroyed, and when it is destroyed, the axon here is dying, and afterwards this neuron will die. So there are several leukodystrophies, and adrenoleukodystrophy is one of the most frequent one, and it's an X-linked disease, means it's linked to the X chromosome, so it's mainly boys that are affected, children and adults. And you see here the frequency of the disease, 1 on 17,000. That doesn't mean a lot of things, of course, but it means, like, for example, in France, we have around 40 families. I say families, not patients. That means several patients in one family. And to give you another view of what it is, the frequency, it's not such a rare disease. We have about 1 new patient every month. So it's a rare disease, it's an orphan disease, but it's not such a rare disease. There are 2 forms in the disease, one which is called the cerebral form, cerebral adrenoleukodystrophy, which is the destruction of the myelin, and as I just showed before, with this terrible, devastating evolution in something like 2 to 3 to 4 years. Mainly in children. This is the repartition of the disease with age, and you see that cerebral ALD, so that happens mainly in children here for like 50%. And the adults, when they didn't have the cerebral form in the childhood, the adults then will develop the second form of the disease, which is called adrenomyeloneuropathy, and which involves the spinal cord and leads to this very severe motor deficit with paraplegia and wheelchair in adults by the age of 40 to 60. And 1/3 of these adult patients will also develop cerebral ALD, so with this very severe evolution as you see here. So you see that cerebral ALD happens in about 65% of all the patients, and with this very severe phenotype. So this is the kind of evolution we have again, and at the beginning, this is very important, because at the beginning here, where you have signs on this MRI, here this is not normal at all. You have already destruction of the myelin, but here you don't have any clinical signs. And this is extremely important, because you can understand that at that point, if you follow the patient, because there are other patients in one family, so you follow a child that has no signs, you do MRI every 6 months, and you see these signs, then you know that you have to treat them very rapidly. Otherwise, it will go through these different steps, and at this point, the disease will be terribly severe, with visual deficits, auditive deficits, seizures, motor deficits, and finally vegetative state or death. So what I want you to understand is that it's very important to follow the patients when you have identified a family, because then you can do this MRI every 6 months and you can see if these signs appear, and then you have to make the decision of treating, because at that time there is no clinical signs, so it's not on the clinics that you can make the diagnosis. So, and this takes about, as I said, about 18 months until you have this very severe evolution. So what can you do? What is the treatment for that terrible disease? It's bone marrow transplantation. And bone marrow transplantation, so the transplantation of your blood cells, stem cells from the bone marrow that can reconstitute the hematopoietic system. And how does it work? Then doing bone marrow transplantation can cure a cerebral disease. It has been used like in the 90s to treat like mucopolysaccharidosis that you've heard before, because these diseases were due to, as I said, Fatima, to the deficiency of enzymes, and these enzymes are secreted, and bringing cells from the bone marrow of a donor who is normal and express normally these lysosomal enzymes can bring this enzyme into the brain of patients. And based on this knowledge and the results of bone marrow transplantation in different types of mucopolysaccharidosis of other diseases, like Gaucher, for example, or Hurler disease, there has been a first transplantation in adrenoleukodystrophy. At that time, we didn't know the gene. The gene was not cloned. We didn't know what it was doing. And so only based on what it was able to do in other diseases, the first transplantation was made, and it was a success. We were very, very lucky because it was a success. Why do I say we were very lucky? Because now we have cloned the gene, and now we know that it is absolutely not an enzyme that is missing, and that it had no reason to work in theory. So if we had known the function of the gene at that time, nobody would have ever done bone marrow transplantation. This is again something I wanted you to know, because it shows you how medicine is not something straightforward. It can take different kind of roads, which sometimes are very complicated. So coming back to the patient, the first boy transplanted here, the first one, had these lesions in white again here that you see. And this is 2 years after transplantation. You see that the lesions have regressed here, for example. And now he's an adult and is perfectly well. So based on these results and these positive results, there has been more than 200 patients now transplanted in the world. So coming back to the question, how does it work? Since I didn't put you slides on the protein that is coded by this gene and what can be its function and everything, but just how can it work? Actually, it works because, again, this is the neuron, this is the myelin sheet with the axon inside that is protected by this myelin sheet. This is the blood vessel that goes to the brain, and this is a cell called astrocytes. And this little cell here is called microglia. And microglia is, when you do bone marrow transplantation, you bring cells from the blood that come into the brain and that differentiate into this microglia. And correcting this microglia does cure the disease. So there is a defect in this microglia in adrenoleukodystrophy. But still now, we still don't know exactly why this microglia is not functioning properly in the disease. However, replacing this microglia, thanks to the bone marrow transplantation, we can cure the disease. So this is the important thing to know, even we don't know exactly how it works. So again, microglia microglia originates from the bone marrow stem cells. So we have stem cells in the bone marrow that are able to give all the cells that you have in your blood, the red blood cells, the white blood cells, the lymphocytes, the granulocytes, everything. And from this monocytic lineage here, giving macrophages, some of these cells go to the brain, cross the blood-brain barrier, and differentiate into these cells that I called microglia. This is how it works. So bone marrow transplantation is efficient. It has been performed in more than 200 patients now in France and in US especially. But it leads to very severe complications, as you may know. For example, graft versus host disease. And finally, about 20% of children that receive bone marrow transplantation from a non-related, not fully compatible donor die from complication of the treatment. And in adults, it's much worse, since it's about 30 to 40%. So this is the reason to find another way to treat the patient. And the other reason is that not all patients, of course, have a donor. So based on that and based on efficacy of bone marrow transplantation, we have decided to try to do gene therapy. The gene was cloned, so we start exactly when the gene was cloned, and it was in 1993. So we could manipulate the gene, as you've heard about from this morning. And we thought that we could correct the own hematopoietic stem cell from the bone marrow of the patient, put back the right copy of the gene, and put back the cells into the patients to cure the disease. So no complication due to transplantation from donors, no graft versus host disease or whatever, and more simple treatment theoretically. So this is what we have tried to do: correct bone marrow stem cells from the patient with a viral vector and do autologous transplantation. So I've put here the tool, the viral vector. So this is the answer how. Today there is nothing that works better to put a gene in a cell than a viral vector. You've heard about it. It's the only thing that we know how to do very efficiently. So it has complications. It's not fully efficient, but we can do it. So I've put several slides. You know that we replace the genes that are inside the virus by the therapeutic gene. We take out all what is bad, and that could be infectious for the patient. And we use that as a horse, a Trojan horse, to get into the cells and give the gene to the cells properly. This is another way to see it. This is, you see an electron microscopy, so these little viruses, and you can see how they can go through the membrane and give their genetic material into the cells, so bring a therapeutic gene into the cells, like you see here. Another way to show you, you replace the genetic material of the virus by the therapeutic gene, so it's inside now the modified virus that can go to the cell and be expressed by the cells. So do the job, replace the missing protein. So which virus to use? So this was a very important question. Coming back to what we've done, so it was in the late 90s. So we used the retrovirus because the retrovirus get integrated into the cells. And if you want to replace these microglia cells coming from the bone marrow cell, this stem cell here, you need to replace it stably, so because this cell will divide into all the different cells, and you have to be sure that the gene that you want to put inside is going to stay there, and during the many divisions of the cell will still be there to do the job in the different cell types, and particularly in the microglia. So you need to have an integrative vector, and the only vector that are integrative that we can use are retroviruses. But we did this work for 10 years with classical retroviruses coming from mouse, and it was working, but it was working very badly, very inefficiently, and so we thought we could never ever have a clinical benefit in patients with that. So we had to wait until this virus from AIDS, the HIV virus, was modified to be able to be used as a tool for gene therapy. And so at that time we moved to use that vector, to thinking that we would be maybe much more efficient to do gene therapy for our patients. So this is roughly what it is about. We take the cells from patients. We take them, the hematopoietic stem cell, actually very simply from their blood. We put them with the virus that have been engineered to express, to carry the ALG gene. We put them all together, and finally we have genetically modified cells that we hopefully can put back to the patients. So we have spent, of course, years showing that this is feasible, that this is efficient, having like more than 50% corrected cells in that bulk here, and that it is well tolerated for the cells with no expected complication than if we would use that in vivo. But then it was a very long road, because I come back to the beginning of the story, 1993, when the gene was cloned, and you see that we were only authorized to do the clinical trial in 2006, meaning that it took a very long time to be able to do that. Why? Mostly at that time because we had to do a lot of work to be authorized by the regulatory agencies to use for the first time this HIV virus to go into clinical trial in patients. And finally we were approved in 2006, but you see that we had a lot to do. So 2006 first clinical trial, and I'm very happy to put that today, because finally 2013 the new trial with more patients treated. So this is a very good news, of course. So sorry, I left this slide in French. So this clinical trial was for patients with cerebral ALD, so patients that do need hematopoietic cell transplantation. So remember the slides with the leukodystrophy evolving, and I said at the beginning you have no clinical symptoms. So this is at that time that we do take the decision of bone marrow transplantation. So these patients are this kind of children with no donor for bone marrow transplantation. So their cells were purified, were transduced with the lentiviral vectors, and were frozen until we do all the safety assays, and the regulatory agency asked us a lot of safety assays to be sure that we wouldn't get the AIDS to the patients. Actually, this was not a concern for us, because the virus was so much modified that he had nothing left from HIV, and we really didn't think we could even ever give AIDS to the patient. Our main concern has been this vector is going to integrate into the cells, and we know from previous trials from the children with immunodeficiencies that there have been concern, there have been leukemias in some children treated with integrating vector. So we were very cautious to follow the patients and see whether there is any concern about HIV able to give also this kind of complication. We did something important. We took out their bone marrow cells, doing what we call bone marrow conditioning with chemotherapy to take out of their bone marrow, so the cells we would put back would really replace the system and would lead to a beneficial effect. So this is again how it is. We take the cell, but this is now the real life. This is what we've done in patients. So this is the patients. We take their cells out, their hematopoietic stem cells out. We transduce them with a vector, but now it's a vector from clinical grade, of course, which has a lot of quality specificities. It's not in the lab. We do all the safety assays, and then we put them back in the patients. And the good news is what you see on this slide, and for me it's the only slide that counts to show you the details of the scientific thing in this trial, is that all these blue things are cells. And you see that you have blue things with no red, which are the cells that do not have correct ALD protein expressed, the cells from the patients. And the cells which have the red dot are those who have the protein that is expressed thanks to this HIV recombinant vector. So this is what you see when you take a blood sample from the patient and you put them under the microscope, and this is what you see. And this was really a great success and a great, I must say, happiness for everybody to see that. So just that to tell you that it shows you that we have now 6 years follow-up in the patient, and when you look at that over time, you see that we have very stable results up to 6 years after the treatment. But you see also that this is a percentage of cells in the blood. So the percentage of cells at the end is quite very low. It's around 10%. And I must say that if we had known before starting that we would have 10% only cells in the blood of the patient that are properly corrected, we would never think that we would have a clinical benefit on the neurological disease. But actually, we have a beneficial effect on the neurological disease, and the beneficial effect is absolutely comparable to what we have after transplantation from a donor when there is no complication like graft-versus-host disease. So I'll show you in a moment the MRI, but what we have after transplantation from a donor is that the disease continues to aggravate, so the white in the blood, in the brain continues to aggravate for a few months until we have stabilization and full stabilization of the disease. And it's exactly what we have in the clinical trial of gene therapy. This is an example of the two first treated patients. You see here this one, the little white dots here with this leukodystrophy lesion that have appeared. So this is just before the treatment. This one has more severe lesions you see here, particularly on this section. And this is 12 months after gene therapy. You see that it has aggravated, clearly aggravated in both of them on the different images. But then it stabilized, and it stabilized up to now. So we do have really a stabilization of the disease, and we do really think now that after 6 years there's no reason why it would change. I don't go into details about the clinical evolution of the patient, just to tell you that 3 of them have really stabilized. The 2 first treated patients have a normal life, normal social life, normal familiar life. They go to school. One has some attention difficulties and is in a special school. The other has lost a part of his vision because of the initial evolution of the disease. But even if he has that, he goes to school and he progresses normally. The last one is the one that has stabilized the more rapidly. We have only 2 years follow-up, but up to now is very well. He has absolutely no clinical signs. The third one has a very much less good result. We think it's not a success because he has, of course, he has stabilized, but with very, very severe lesions, and is not able to go to school now. Is not able to talk, and so we really feel that is not a success. But it is also something that you see sometimes in some patients after transplantation from a donor. What you have to keep in mind is that it's the evolution of the disease. This is a non-treated patient in the past that had no donor for transplantation with this terrible evolution in 24 months. And this is the first treated patients that are now stabilized. This is 36, but it's the same at 64 months now. So this is the result of the clinical trial. So what are we going to do now? As I said, we want to treat more patients because 4 is only 4, and we need to prove that this is really an option for patients to choose between the transplantation with an unrelated donors with a risk of complication and of death, as I said, in about 15 to 20% in children, and treat adult patients because, as I said, in adult patients, the mortality risk with transplantation from donor is much higher, like up to 40%. So we really think that it is an option, but we do need to prove that it is one by treating more patients, and this is something that that is done currently, starting in the US and in Europe soon with the Bluebird Bio company. And the other thing, and I won't tell you about today, but this is of course something we have in mind also, is to treat the adult form of the disease because it's very severe and it leads to wheelchair, these men. So we really want to target the spinal cord. And targeting the spinal cord is another story with other vector, and you've heard about AAV vector with Fatima, and it's exactly what we plan to do for that. So I'll stop that. I hope I wasn't too long and that you could understand part of what I said. And please, Juan, go back into the slides if you think you have to do it. I just want to thank, where is he? You know what? I don't see, I missed the slide. I missed the slide. There is no, not the lab on the slide. So I just want to thank Patrick Aubourg, who is the PI of this clinical trial and who's done all the work. He has cloned the elder gene in 1993. He's following all the patients in France and a lot of patients worldwide, and he's really the one that has done everything in that story. And I also can thank the group of Necker who has helped us a lot for transducing cells, particularly Sabina Athenbey, and the group of Heidelberg who done a lot of work on the follow-up of the patient, showing that we've done something sure and that were very well tolerated with no problem of leukemia. And I wish to thank the association that have helped us a lot, particularly the AFM and ELA, which ELA is European Leukodystrophy Association, and has worked a lot with us for many years. And ELA in Spain is also very active, and I must say that the 3 first treated patients were from Spain, and we were very happy to work with ELA. Also, of course, I wish to thank Bluebird Bio, who is now doing the next trial and, of course, will help to put that drug on the market in a few years. Thank you very much.
Thank you very much indeed, Nathalie, for your clarity and also on behalf of the patients for your work. Como os decía al principio, han sido las primeras evidencias de eficacia clínica en esta patología y ha habido muchas preguntas que resolver y decisiones muy complicadas que tomar, como por ejemplo la necesidad de hacer un acondicionamiento con quimioterapia previo a la infusión de estas células sin tener todavía la seguridad de que iba a tener beneficio clínico. Y también nos aclara una cosa muy importante que ha surgido en una pregunta anterior. ¿Es necesario que las células que sean diana para corrección genética sean las células que sufren la enfermedad? Y este es un claro ejemplo de que no es así. Puedes corregir células madre de la médula ósea para tratar una enfermedad neurodegenerativa que se produce como consecuencia de la afectación de neuronas. El ejemplo es inmejorable. Bueno, ahora podéis hacer preguntas, como os decía, las podéis hacer en castellano. En la medida de lo posible nosotros las podemos tratar de traducir para que las pueda entender Nathalie. directamente en inglés, en francés. Si queréis que os aclaremos alguna cosa de la presentación, lo podemos hacer. Tomar la libertad, no hay pregunta que no sea interesante, sobre todo en tratamientos que son tan complicados como este. Sí, adelante.
Nathalie, thank you very much for your beautiful presentation. It's very encouraging for us to see what's happening. As you began saying that in medicine things are not straight. It's a very good thing now to know that when you have a bone marrow transplantation, you can cure something you don't know what's happening even.
Exactly.
No? So in this place, in this moment, when we have these types of things like vanishing white matter, the other man told us, no, or CAT syndrome, and we cannot do anything else. Do you think in this moment it would be not so bad to try to do something like bone transplantation in these patients?
It's a good question. The problem is to treat patients at the very early time when the disease is not too evolved, because then it's too late. We don't know how to repair, you know. We know how to stop the progression, but we don't know how to repair myelin. This is something a lot of people work on, of course, to repair the myelin and to replace, eventually to replace neurons. But today, this is something we don't know for clinical application. So we have to, that's why I emphasize so much, we have to make the diagnosis very soon to be able to do whatever, but very soon, at the very early stage. And as I showed, doing bone marrow transplantation is a very slow process to go to the brain and to replace the microglia in the brain. So we have to anticipate that after bone marrow transplantation, it will continue to aggravate for several months until you have the stabilization. So, but it could be an option, of course, even you don't know the gene implicated in the disease, of course. Mm.
Yes.
I understand what you mean.
You can get to know it, but it takes time. It's not so easy.
No.
So you get after 2 years, if it's slow.
It's
looking
like,
after.
roughly it's like 1 year, let's say 1 year.
When you get to the factor,
yes.
no, transcription. But the thing is, if in this case, the stress of the transplantation is going to be worse for the patient.
That's a problem.
Yes,
and
also,
also
no?
the problem is that you, the risk of bone marrow transplantation is not zero, as I said. And so you have to balance the risk of the mortality risk of the transplantation and the risk of the disease, even if it's a terrible one.
Thank you very much,
You're
Nathalie.
welcome. Yes.
For
Hello.
your address. Hello. I have a patient with CAT syndrome. I want to know which is the difference between adrenoleukodystrophy
Yes.
and leukodystrophy. Simple.
Other leukodystrophy? Yes.
And this solution is useful for this case.
What is the disease you have?
CAT syndrome.
Yeah. Okay. So no, it's not the same. This is adrenoleukodystrophy is a disease of the. It's linked to the ALD gene is in peroxisome, and it works on long chain fatty acid. So it is not the same kind of disease. I don't know what are the results of bone marrow transplantation for your own disease, but I can check and go back to you with the answer if you through the patient association and
Then I asked
yeah.
my
Yeah.
others.
Yeah.
Thanks.
Of course.
Okay, one last question. Okay.
Hi, I have a question regarding the transplantation. Since you said that the patients were sensible to the transplant, I was wondering, was it maybe possible to use the cord blood cells that are frozen before the freezing, after the thawing them, transfer the cells with a gene, and keep a bank of the cells already expressing the gene of interest in your case,
Yes,
and
of
then
course.
use it in the future?
You mean
as a
taking the
medicinal drug
cord
already?
cells from the patient, you mean?
Not from the patient, the cord blood bank,
Yeah?
where donors give the
Yeah.
cord blood cells, transfer these cells with your gene of interest, and keep them frozen as a medicinal product to treat the patients in the future. So you overcome the problem when you need to extract the cells from the patients.
Yes. So first there are cord blood transplantation, as you probably know, from donor cord blood. Huh? Okay. But of course, if we have the diagnosis when a child is born, of course we can take the cord blood and say, if once we can cure the cells, we will put them cells back there. Right. Of course, but then you have to know that the child has the disease, because today you don't take cord blood from any child that is born.
Unfortunately,
that is the problem,
Yeah.
of course. But let's say if there is a child in a family with ALD that is going to be born, and the parents don't want to have the prenatal diagnosis and want to keep the child even if he has the disease, which happens, then you can, of course, as you said, take the cord blood and think about in the future when the treatment is approved after the next trial, hopefully, then we can do what you said.
Actually, my point was using what is already in the bank, an allogenic transplant,
so
since they are very low in the...
It's not necessary, since then it's normal cells, so you just do a transplantation of cord blood, not manipulating them. No use.
Okay.
No use.
I'm sorry, but
Okay.
I should stop because it's getting late. Thanks a lot again, Nathalie.